Okay, good morning, everyone. My name is Sabrina Sholts. I am a new curator in anthropology, and I'm going to talk fast. Okay, so I changed the first word of my title. Sorry. Really what I want to talk about, the concept that I wanted to discuss today is paleotoxicology. I define paleotoxicology simply as the study of toxins in pre-industrial populations. This is a unifying theme of a lot of the projects that I'm doing right now, and so I wanted to talk a little bit about 3 of those in order to give you a sense of what I'm doing, where I'm headed, and I'm going to actually then start with where I've been. On the background of this slide, you can see Santa Barbara, California, which is where I did my PhD. And off the coast of Santa Barbara, you see the California Channel Islands, which were the focus of my dissertation research. These islands were occupied by people for about 13,000 years, and I was studying changes in craniofacial morphology over time. I was using collections of archaeological skeletal remains that were in museums. But if you go out to the islands and you actually experience this place where these people lived, you can't help but see so many ways in which human biology may have responded to this environment. And so what I see here and think about the most is pollution. Actually, everywhere you look on the beaches of the islands, you see petroleum, which seeps naturally from cracks in the ocean floor and floats to the surface. And the oil platforms that you see in the Santa Barbara Channel, those signify that this is a very rich area for petroleum seepage. So if you're an archaeologist on the islands, you see petroleum everywhere in the archaeological record because people that lived on these islands used it extensively in just about everything that they made, including containers for food and water. And so I became very interested in how the chemicals in petroleum, known as PAHs, may have actually impacted human growth and development and health over time in the past, because in the present, we know that these chemicals are highly hazardous. PAHs are actually at the top of the list of priority pollutants that are regulated by the EPA, and they outrank a number of man-made synthetic chemicals that are highly persistent in the environment. And so some colleagues and I proposed that increased reliance on petroleum may have actually been a factor in declines in head size and stature and health that we see in the skeletal collections from these islands. However, there are a lot of factors that could contribute to and explain these patterns. And so in order to adequately test this hypothesis, we need to do a lot more work. It's very difficult to quantify exposure in the past and to identify effects of exposure when really all you've got to work with are bone and teeth. And so I'm just going to share with you a few different ways that we're approaching this problem and that I think are promising avenues for paleotoxicology in the future. Our projects deal a lot with metals because metal does accumulate. It's stored in bone tissue. And also there are a number of potentially very serious effects of metal exposure, such as neurodegeneration and endocrine disruption. And I think as we understand more about these effects, that metals are going to go up on this list. At the bottom of the list, under the metals, what you see is a chemical, a synthetic chemical known as TCDD, which is probably best known as the chemical that was used in the attempted assassination of Viktor Yushchenko in 2004. And besides the chloracne that he experienced as a result of this episode, there are many other effects that can occur from TCDD exposure at much lower doses. It's the most potent chemical in the dioxin family, and it can serve as a model compound for understanding mechanisms of dioxin-like compounds such as PAHs. So we used TCDD as a model compound to understand dioxin-like effects in cranial growth in laboratory rats. Of the model organisms that are used to understand human development, rodents provide probably the best reflection of human head development. And so it was a nice way to actually look at dioxin-related effects in a controlled dose-response experiment. In a sample of 2 rat strains, one that had a mutation that provided resistance to TCDD and the other that had a natural sensitivity to dioxin, we found that craniofacial form was altered by exposure to TCDD during adulthood. What we saw most significantly was a shortening of the face, which is because anterior facial growth is really the dominant process that continues to occur in adulthood. And so the results of this study show that timing is a critical factor in toxicity. Had Viktor Yushchenko been a child or an infant when he received that dose of TCDD and not a grown man, he probably would not have recovered or even survived. In another study where we're continuing this exploration of the timing effects in toxicity, we're comparing the adult exposure sample to a sample of rats that were exposed before birth and immediately afterwards through the dosing of the mothers. And so on the left, in the blue, you see the shortening of the face, but on the right, in the red, in the prenatal exposure sample, what you see is really that the cranial dimensions haven't been affected in the adult rats, but there's more of a movement of the intersections of the sutures. And this is probably because these sutures fuse during the first 2 weeks of life, and high concentrations of TCDD in the mother's milk provided probably the highest level exposure that was received by the nursing offspring. And our results suggest that TCDD may have some sort of role or function in the expression of a non-metric cranial trait known as the interfrontal bone, which you see below. And actually, that's very interesting because non-metric cranial traits are generally perceived to be in presence or absence a hereditary trait, or they might be affected by mechanical function, but certainly not something that you'd expect to see with toxicity. So this should shed more light on looking at cranial remains in the future. And so I'm actually going to very quickly say that another avenue for looking at paleotoxicology is genetics, where our colleagues in Sweden have actually found that you get these higher frequencies of this genetic variant that provides some resistance to arsenic when you're living in an arsenic-rich environment. And we'll be exploring the antiquity of that human adaptation to chemical exposure. And then finally, on the Channel Islands, going back there and looking at some sort of environmental monitoring. We're going to be analyzing trace elements, concentrations of metals in fox bones, which will allow us to understand how these levels have changed and may have been caused by and also influenced human populations on the islands. So, thank you.
A question for Sabrina. You ended your talk with a tantalizing reference to evolution and arsenic. Yeah. Could you say a little bit more about that, please?
Sorry, I don't see what this are, the questions.
Over here.
Oh, hey, yes, of course. Absolutely. So, there have been a number of studies that come out from a group of scientists in Sweden based in Uppsala and Stockholm, and they have found that focusing specifically on the Andes, in the Altiplano region in northern Chile and in western Bolivia, that the living populations in this region, that they actually they show these higher frequencies of these genetic variants that do allow them to metabolize arsenic more efficiently. There are changes in the pattern of methylation, and that's actually conferred some protection against the arsenic in the environment and allow them to live there for so long. And so really what we want to do, because it is quite a novel idea that this arose in these populations, and we do have some archaeological evidence showing that the mummies, the Chinchorro mummies, for example, in the same region, that they are showing elevated concentrations of arsenic. And so the exposure was probably there. But we don't have any ancient DNA to actually prove that we had similar frequencies going back into the past and when that may have actually began. And so we have started to sample some mummies from northern Chile to start this work, and we'll be continuing on with this, hopefully, and looking at other regions of the world to better understand this question and shed more light on the history of this adaptation.
So, Sabrina, I have a question for you as well that's sort of following on Kay's question. So, are there any other potentially morphological effects that you can imagine from the toxins that you're studying, whether in a rat model or something else? Are there predictions that you can make about that?
About any of the toxins? Do you mean, for example, the petroleum, like on the Channel Islands? There are certain, diseases that have been associated with exposure to certain pHs, and polycyclic aromatic hydrocarbons is what we're talking about, and there are a lot of them. It's a huge class of chemicals, and so when you're talking about petroleum, you're talking about a whole mixture, and you can look at, specific effects of single compounds, such as naphthalene. And this is something that my colleagues and I have been looking at, that naphthalene in particular, and that's the kind of chemical that you get in mothballs, that's been associated with anemia, hemolytic anemia, for example. And so that's simply one example. I think with the hydrocarbons in general, they act the way a lot of harmful chemicals act, in that they just disrupt growth and development. And so it's going to be a systemic effect more than something localized or specific. But it certainly, it would be difficult to characterize anything as a smoking gun. Nonetheless, I think that we can at least build support for this hypothesis and really think about these health factors that are certainly important and relevant today, and are equally so, would have been in the past. So.